Hi everyone, thank you for being here today. I'd like to thank the DDW committee for this opportunity, and I have no disclosure. By this point, we all agree that diabetes should no longer be framed simply as a disorder of elevated blood glucose. Contemporary evidence supports a system biology view in which diabetes reflects dysfunction across multiple interacting organs, including the pancreas, liver, adipose tissue, and increasingly in the gut. In this framework, hyperglycemia is a downstream clinical phenotype rather than the full disease itself. These figures illustrate diabetes as a multi-organ network disease. At the center is the systemic metabolic dysregulation. Insulin resistance, inflammation, and dysglycemia are not an independent process. They form instead a self-reinforcing cycle connected to multiple organs. And alterations in one organ or process propagate through the network, amplifying dysfunction across others. One of the most conceptual advances in diabetes research is the recognition that type 2 diabetes it's not a single biological entity. Patients who meet the same diagnostic threshold might actually have fundamentally different disease drivers. If we accept that type 2 diabetes is a heterogeneous disease driven by distinct biological pathways, the next question becomes, what are the upstream modulators of this heterogeneity? One increasingly important player is the gut microbiome, which serves as an interface among the environment, metabolism, and host signaling, including inflammation and immune regulation. Mechanistically, the most plausible pathways include altered short-chain fatty acid production, changes in bile acid transformation, changes in epithelial barrier function, inflammatory signaling, and others. With that said, the gut microbiome is highly relevant biologically in the context of diabetes, and given the proposed molecular mechanism by which gut microbes can influence diabetes outcomes, it is clear that the small bowel microbiome is the main player here. So glucose absorption and regulation, GLP-1, GIP, and insulin regulation, immune response, and many other processes classically involved in diabetes are mainly affected by the small bowel homeostasis, and therefore its microbiome would have a big impact here. In fact, small bowel bacterial overgrowth, or SIBO, one of the most well-known microbiome-related conditions, happens in the small bowel, and the strongest biological rationale lies in the diabetes-associated neuropathy or delayed transit. In this context, the small bowel might become more permissive to microbial overgrowth and symptoms generation. Well, as I walk you through our data today in the next few slides, I will bring this theory back for further discussions. So given the importance of the small bowel and its microbiome in the context of diabetes and SIBO as one meaningful gastrointestinal comorbidity in this subgroup, we aimed in our study by using quantitative metagenomics sequencing to characterize the small bowel microbiome in type 2 diabetes and SIBO and their relationship to fasting glucose levels. For that, we select subjects from the reimagined study at Cedars-Sinai. Currently, we have the largest collection of small bowel aspirate for microbiome studies in the United States, along with blood samples, biopsies, and medical records. We recruited subjects undergoing standard of care upper endoscopy without colon PEP. We collected the duodenal luminal aspirate using a double lumen protected catheter to avoid cross-contamination with other sites and blood. And aspirates were immediately processed after collection for SIBO diagnosis on McConkey agar. DNA was extracted, and we performed quantitative whole genome sequencing for metagenomic analysis on a NovaSeq platform. And this is the demographics of the cohorts for this study. We did observe, in fact, that fasting glucose levels was higher in subjects with type 2 diabetes compared to those without type 2 diabetes. And we also observed differences in sex and BMI. And these two variables were taken into consideration during the analysis of the microbiome. So in fact, the prevalence of SIBO, now detected by a direct culture on McConkey agar, was higher in subjects with type 2 diabetes, and the number of gram-negative bacterial colonies on McConkey agar was also higher in these subjects, even when SIBO cases were omitted. We also observed a significant drop in duodenal microbial diversity. When we broke down our subjects by both conditions, the co-occurrence of type 2 diabetes and SIBO was associated with the greatest drop in microbial diversity in the small bowel. And considering what we know about SIBO, two key players immediately come to our minds, Escherichia coli and Klebsiella pneumoniae. So we decided to take a look at their duodenal absolute abundances across these groups. And the results with E. coli were revealing. So subjects with both conditions, with negative for both conditions, had the lowest absolute loads of Escherichia coli in the duodenum. And positivity for type 2 diabetes alone was already associated with increases in this load, followed by SIBO alone. Co-positivity for type 2 diabetes and SIBO were associated with the highest E. coli load, even higher than SIBO alone. Next, we decided to take a look on the fasting glucose levels in these subjects. And the levels of glucose were associated with duodenal lows of these species as well. When we broke down our glucose levels by percentiles, higher tiles were associated with higher duodenal loads of Escherichia coli. And the prevalence of SIBO was also significantly higher in the higher tile categories, including the second percentile. And this event was not associated with higher prevalence of type 2 diabetes, indicating that SIBO associations with higher glucose levels are an independent phenomenon and may perhaps precede the establishment of diabetes. With that said, I would like to propose another mechanistic plausibility for SIBO and diabetes, and suggest that change in the microbiome, specifically related to higher loads of Escherichia coli in the small bowel, may lead to glucose homeostasis disturbances and increased risk for diabetes. But obviously, more studies are needed to understand this possible connection. We also examined the entire duodenal microbiome composition subject with type 2 diabetes, and several Streptococcus species were enriched in the duodenum of these subjects when compared to controls. We also observed a higher absolute abundance of Fusobacterium nucleosphorum, which was also related to fasting glucose levels. We had some limitation in our studies. A small number of subjects for further stratification based on the medication and control status. The diabetes status was self-reported, meaning that we might have some patients with prediabetes or false negative as part of the non-type 2 diabetes group, and the absence of more sophisticated tests to evaluate the diabetic state, insulin response, and diabetic course. So in conclusion, we confirm that there is a strong link between type 2 diabetes and SIBO, but now SIBO diagnosed by direct culture on McConkey agar. Subjects with type 2 diabetes have a substantial loss of duodenal microbial diversity. And co-positivity for type 2 diabetes and SIBO is related to enrichment of Escherichia coli in the duodenum, even higher than SIBO alone. Escherichia coli duodenal loads correlate with fasting glucose, and fasting glucose levels are linked to SIBO positivity. Several Streptococcus species and Fusobacterium nucleosphorum are also enriched in the duodenum with subjects with type 2 diabetes. And Fusobacterium nucleosporum's absolute abundance also correlates with fasting glucose levels. So diabetes heterogeneity is not only genetic or metabolic, it's also shaped by the gut ecosystem. Thank you. I would like to thank the MAST team and all the participants in our study. Now I'm open for questions.